Hi Bio4ers, and welcome to Neurons Part 2. So before we go on to those glial cells that I had advertised, we're first going to look at a little bit more about neurons themselves. Neurons as a function are excitable, right? They have the ability to respond to a stimulus. You can excite this whether it be by light in the eye or by sound in the ear. Something is going to excite this neuron, and it's going to be able to conduct some sort of impulse. And the light in the eye is going to conduct an impulse, come into the eye, come into the brain, and form an image, for example. Okay, so it has that excitability, conductivity. And three, some neurons are able to actually secrete neurotransmitters. So they are called neurosecretory cells. They're able to secrete. So they secrete neurotransmitters, and in that way, they stimulate an adjacent cell, for example. So classic functions of neurons. Let's look at what they look like anatomically, morphologically. So there's three types. You would have a sensory or afferent. So in order for us to remember this, afferent and efferent, never make a mistake with that. Afferent, A for arriving at the central nervous system. A for arriving. So this is what one of those sensory neurons would look like. It could have these dendrites and then feeding into where the cell body is, and then one long axon. Or it could have dendrites feeding into the cell body, but do you notice how the cell body is in the way here as opposed to off to the side? These are both sensory neurons. So they will detect temperature, they will detect light, pressure. If something touches you, you know that's stimulating one of these pressure receptors. And then all the special senses that we'll be looking at, which are exactly how we hear and how we see, and we'll be looking at that in future lectures. Now, the interneuron is entirely within the central nervous system. Many of them are really loaded up on their dendrites. Look at this, because they're going to connect with various other cells. And they are going to connect those arriving and those exiting. In other words, those arriving, afferent, and exiting, efferent. Arriving, afferent, exiting, efferent. So here's the motor neuron that you drew in lab. It's going to connect that central nervous system to those effector organs or muscle or glands. So it had one big soma here, or cell body, its dendrites, and one large axon. Okay, let's put all those three together now in one slide. And this is what it would look like. If this were a picture of the spinal cord, central nervous system, correct? Well, we would have then a sensory afferent arriving, afferent coming in all the way to the central nervous system. There, it's going to meet up with an association neuron or an interneuron and then connect to an effector, to an efferent neuron, one that is actually exiting. Now, when we think of this, can it really be three? As little as three, or is this is just a diagram to make it simple, but it's not really like this because it's millions before it comes back out again? And the answer is no, they can be three. It can be one neuron coming in, one connecting, and one exiting. And this would be the example of a reflex. So in a reflex, you don't have these neurons connecting up. I'm going to draw it up like this, up to the cortex of the brain. That would be up here. It doesn't travel up there. It just enters into the spine, connects, and goes right back out that same place in the spine. Most of these coming in, connecting, and exiting are not like this. Most of them will come up to the cortex of the brain, interpret that, and then will come back out. And so it would involve 4 or 5 or 6 of these neurons, depending. And another really important thing that confuses so many anatomy students, notice where the cell bodies are. Okay, so let's notice this one. Cell body, central nervous system. Cell body, central nervous system. Well, this one that's afferent, the cell body isn't quite in the central nervous system, but it's really close. It is never out here like this. Never. It doesn't happen. It's going to be in this location, which is called a ganglion. So a ganglion is a location that's harboring, hiding out these cell bodies. And that tends to be very close to the spine. So every single cell body that you have is either in the spine, in the brain, or really close to the spine. You do not have a cell body halfway down your arm. You do not have a cell body halfway down your leg. They're just not there. What you would have is the axon or a very long dendrite, right? This is a very long dendrite that comes all the way to here, and then from here on, it's an axon. But you don't have a cell body in your toes. Remember, you want to protect those. If you were to damage the cell body of a neuron, you can't replace it. If you were to damage here a neuron like this is going to actually grow a new extension. So if I were to make this in blue, this would sort of extend out like this and form new extensions in that area and do its best to connect to what it connected before. But if I were to damage a cell body, that is the end of that neuron. So we're going to protect them. We're going to put them safely away in the central nervous system, or we're going to stash them in these little pockets called ganglia. Okay? And if I can backtrack a little bit for you guys and show you one of the slides that I showed you last time, where here you see, here was the spinal cord, central nervous system, and then next to it, look, bump, 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 bump. These are all ganglia that are very close to the spine. Okay, that's the only other place that you would have cell bodies. Okay, what we want to now do next is look at the glial cells. Excuse me. And I'm going to leave that for part number 3. Let's stop there. Thanks for coming, guys.